Awesome. Okay, well, let's get going. Good evening, everyone. Thank you so much for coming. We're really excited to have Antonio Rosas here with us tonight. And it's through the Spanish Embassy in Pretoria that we've been able to have this collaboration with Origin Center, and also with the Spanish researchers in South Africa as well that have been part of this and advertising the event. So we've had a few years of working with the Spanish Embassy in the past. So really nice that we're able to do this again, even though there's COVID and you're not visiting South Africa. But hopefully, Antonio, sometime you will be able to come to South Africa and join in the fun here and maybe do a live talk at Origin Center sometime. Cool. Okay, I'm just admitting everybody. Okay, so I'm going to pass over to the head of Origin Center. Just now, let me just give you a little bit of the order of events for tonight. We are first we're going to have Professor Amanda Esterhuizen, who's the head of Origins, and she's just going to give a short welcome and then introduce the ambassador here at the Spanish Embassy in Pretoria, Mr. Carlos Fernandez Arias, who's sent a short video message. And then we'll pass over to the man of the moment, Prof. Rosas. And then after, he'll talk for about 45 minutes or so. And then afterwards, we'll have the chance to have a Q&A session. We can write in the chat, or you can put on your sound and your video after the talk, please. Okay, so Mandy, we can pass over to you.
Thank you, Tammy.
Cool.
And thank you to everyone who's taken the time to join us tonight. This is, as Tammy's already said, the third public lecture that Origins is holding in collaboration with the Spanish Embassy and the Spanish researchers in South Africa. I've been so looking forward to this lecture on the evolution and paleobiology of Neanderthals. As indicated by Professor Rosas in his abstract, Neanderthal studies have shaken, and I think in many ways shaped, our understanding of human evolution. And due to the many studies, including, of course, those of DNA, we now think differently about early hominin migrations and interactions. And of course, from my personal sphere of interest, the ongoing reassessment of Neanderthal burials has also set a benchmark for detecting and determining funerary behavior. But let me not get too carried away and allow the Spanish ambassador, Carlos Fernandez Arias, to introduce the speaker.
Cool. Now I'm going to share my screen.
Good afternoon. Once again, the Embassy of Spain is pleased to collaborate with the Origins Museum of the University of Witwatersrand in Johannesburg to make public the main paleontological and anthropological discoveries in my country. After the talks of Juan Luis Arzuaga in 2019 and Dr. Angela Delgado in 2020, this year, together with the Society of Spanish Scientists in South Africa, we have invited Antonio Rosas, Spanish biologist and anthropologist, doctor in biological sciences from the Complutense University of Madrid and director of the Paleoanthropology Group at the National Museum of Natural Sciences of Madrid. He has worked in the Atapuerca sites and has participated in the project of the Neanderthal Genome. Today, he's going to tell us about his findings on the Neanderthals in the El Sidrón cave in Asturias, in the northern part of Spain. The sample of human fossils from El Sidrón represent today the largest and most complete collection of Neanderthal remains found in the Iberian Peninsula, with a collection of more than 1,900 human remains that are corresponding to a minimum of 12 individuals. Thank you very much, and I hope you enjoy it.
Okay, cool. So thank you, Carlos. So I would like to just firstly just introduce Professor Antonio Rosas. I'm sure many of you here tonight actually know a lot of what he's done, but also just to give you a background into his amazing career. so far. Professor Antonio Rosas is a Spanish National Research Council research professor and the director of the National Museum of Natural Sciences in the Department of Paleobiology. Oh, excuse me. I'm having to admit people at the same time. There we go. Antonio obtained his PhD in biological sciences from Complutense University in Madrid in 1992. He held a postdoctoral position at the Natural History Museum in London from 1993 to 1995, after which he returned to Spain to join the National Museum of Natural Sciences in Madrid. He was a member of the Atapuerca research team from 1983 to 2004. Among many other works, he participated as a co-author in the description of the hominin species Homo antecessor. In 1997, as a member of this team, he was awarded the Prince of Asturias Award for Scientific and Technical Research and the Castilla y León Prize for Social Sciences and Humanities. Since 2003, Antonio has been responsible for the study of the Neanderthal fossils recovered at El Sidrón cave, a cave which presents the largest collection of Neanderthals in the Iberian Peninsula. He has also participated in the study of fossil material of diverse origins across Spain and in other countries, including Syria, Georgia, Armenia, and Tanzania. In 2014, he started research work in Equatorial Guinea, focusing on locating sites related to the earliest populations in Africa and to better understand the biomechanics of the first hominins. He's the author of more than 300 scientific publications and 5 popular books. So I'm sure we're in for a very interesting talk tonight. And I look forward to hearing it, Antonio, and you can share your screen. Over to you.
Okay. Thank you very much to everybody, and good evening. First of all, I would like to thank the organizers for inviting me to give this lecture and to participate in this sort of seminars. And it is a very nice opportunity to share some of our findings, research, and interest with you. We are going to talk a little bit about the evolution and paleobiology of Neanderthals, and we are going to have a look from the El Sidrón site, which has been said before, provides us the largest sample of Neanderthals in the Iberian Peninsula, and has become the example, one of the references for the study of this human species in the last few years. There are quite a lot of aspects to treat or to talk about, and I choose for this occasion a few of them. And in any case, after the lecture, we can talk or discuss about these aspects or other ones that could be of your interest. Let's go to start. Asking us.
Okay, so you will have to just share your screen again. Sorry, it went off when I played the video, so just share again.
Okay.
And just while we're doing that as well, if anyone in the audience does have questions along the way, you're welcome to type them in the chat, and then we'll deal with them afterwards.
Can you see the screen?
Not yet.
No. Oh.
So funny how it always works. Everything goes smoothly in the practice.
I should be in. I don't know what's going on. Let me check. It was working before.
Yeah, it worked perfectly.
No idea what's going on. I'm sorry.
Perhaps if you can unshare and reshare.
Yeah.
I'm going to go out and then again try to join the meeting. Okay.
Oh, okay. A little pause. Can all go get a cup of tea or a glass of wine or whatever. Go to the toilet quickly. I find it so funny how this so often happens that you, there we go, he's in here now. Okay, cool. Yeah, that everything goes so smoothly in the practice run, but then when you go live, then all these funny things seem to happen. Okay, I see Antonio is here. Huh? Ah, there we go. Yes, we're seeing it nicely. Perfect. We just can't hear you yet.
Can you hear me?
Yes, I can hear you now.
And the screen is fine?
The screen's good, just got the Zoom thing in front of it.
Okay. Let me. Now?
Okay, over to you.
Okay,
Thank
fine.
you.
I was sweating. Okay, I was saying that we're going to ask ourselves a first question, a general one, for introducing the world of Neanderthals. Who were the Neanderthals? As we know now, Neanderthal is a extinct human species that were living in the west part of Eurasia. Traditionally, we thought that the Neanderthal were especially living in West Europe, but in the last few years, new findings has increased a lot the geographical range of this species. So nowadays, we consider that Neanderthals were a human species extinct that were living in the west part of Eurasia. And traditionally, Neanderthal has provoked a large interest for scientists and also for a general audience, for general people. And we may ask, why to study Neanderthals paleobiology? What is the basis of this great interest that we have in this human species? There are several reasons of this general interest. The first one could be an innate curiosity we have as human beings and as scientists. And also Neanderthal has been chosen as a reference to study the origin of specific human traits. For example, the life history pattern. Comparing our life history pattern or morphology, anatomy with some kind of reference, and Neanderthal has been used as a reference, is a way to explore what make us humans, what make us humans as a unique morphotype, phenotype, and species in general terms. Nowadays, with the influence of our genetic knowledge and the paleogenetic studies, the Neanderthal has revitalized their interest because we are now, generally speaking, trying to understand what is the influence of the genetic influence on our life, on our phenotype, and especially in our health. What is the disease risk that we have inherited from the interbreeding with Neanderthals? We will talk about this interbreeding later in the lecture. And also, there is an historical coincidence between the discovery of Neanderthals as a fossil species in 1856 and actually the recognition of the evolution as a general processes. It was actually in the middle of the 19th century when both the discovery of a human species and the discussion if they were different or they were the same as us, and what is the meaning of this discovery. The first one was in Feldhofer in Germany. And 3 years later, the publication of The Origin of a Species by Charles Darwin that made clear that evolution is a general process in the history of life. And both problems, the explanation of Neanderthals phenotype and the explanation of evolution in general that we can summarize nowadays in the relationship between genotype and phenotype as a specific biological problem. So combining both, I think nowadays the interest of Neanderthals still is alive and quite alive, because actually combining both problems, we don't have a clear answer to the question, what is the causal relationship between the genotype and the phenotype of Neanderthals? In other words, why Neanderthals look like Neanderthals? Still, this question has not a clear answer in science, even 150 years later of their discovery. There has been, as you know, many interpretations about what Neanderthals look Neanderthals, but many of them, or I would say all of them, has been refuted and new ideas has been developed in the recent years, and it's actually part of our research in our group. And the Neanderthals from the Sidrón side has been, let's say, an excuse in this way of speaking to go into the more details of the evolution of the Neanderthals. Nowadays, as I said, Neanderthals are considered a human species, extinct human species, but more than a single species in an isolated way, we think, or we consider, generally speaking, the Neanderthals represent actually an evolutionary lineage. When we speak properly about Neanderthal, we should speak about the Neanderthal lineage, because there is not just one species, but the evolution, the lineage of evolution that gave rise to Neanderthals. In this, on the left diagram, we can see a phylogeny of hominins, and in the circle, we can see at the middle the Homo antecessor species that has been published as a potential or hypothetical last common ancestor of two different lineages. One is given rise to our own species, Homo sapiens, and the other lineage is given rise to the Neanderthals, what we call technically Homo neanderthalensis, with one species before in this evolutionary lineage that we call, some of us, we call Homo heidelbergensis, and recently has been discovered that in this clade, in this lineage, has been another branch that we didn't know until the paleogenetic has clarified the existence of this lineage that we call nowadays the Denisovans, and there is no, formally speaking, there is no Linnean name for this Denisovan human group. The last common ancestor of these two lineages that made the reference for the comparisons of Neanderthals and modern humans has been estimated into 550,000 or 765 years ago, the time of living of this last common ancestor. If we look at this diagram, we can see with these climatic curves some example of the fossils that represent the Neanderthal lineage. Fossil that has been found in Europe. No, I have to say that not everyone will agree with this way of thinking or way interpreting the fossil record, the human fossil record in Europe, because some people think that, for instance, the Atapuerca, Cima de los Huesos fossils are a different group or species from Petralona or Mauer. In our view, this sequence represents the different step of the evolution of Neanderthals, which constitute what I said, the Neanderthal lineage. And if we increase a little bit this range time, we can see here around 49,000 years the sample of El Sidrón. That is the point of reference for our research in the context of the evolution of Neanderthals, of the Neanderthal lineage. If we go into more detail of this general framework, we can see that classically we are looking now at the time interval of the 300,000 years, with the different climatic changes. In blue, we can see the stage that has been cold stages, what has been classically called the glaciations, and in red color is the period that the weather was warmer. In this sequence of time, it has been differentiated, different names for the population of Neanderthal has been found there. First of all, the earlier ones has been called the pre-Neanderthals. Then the first one, there's the early Neanderthals, and then we have the classic Neanderthals until we found this quite important period in prehistory, in the general prehistory, and specifically in the European prehistory, that we call the transition between the middle and the upper Paleolithic, with the substitution of the Neanderthals that were living in Europe and in Western Eurasia by the modern human, the anatomically modern humans that we call ourselves Homo sapiens. This transition is an important, it's a significant point in the prehistory of Europe, and El Sidrón sample is located chronologically just below this transition that occurred approximately 40,000 years ago. Let's move on a little bit on the site itself for the Sidrón. This site is located in northern Spain and is located into a cave. And we can see here a couple of pictures of the cave itself, where there is a very small chamber where the fossils are concentrated in a very really restricted area. We can see in the middle of the slide a picture with the arrow pointing to some fossils that were, in this particular case, in anatomical connection. Here is another view of the main gallery. In the plan, we can see the cave system, where the site is located, and we can see the long gallery, the one that is recorded in the picture, and then in a very small chamber. This is in a larger scale, where the grid has been installed for the digging, for the excavation, and the fossils come actually from this very restricted area. And we have here on the right just an example of one of the most significant assemblage, which belong to what we call juvenile wine. We will call a little bit later on this. More aspect of the collection itself, it has been referred at the introduction that this bone assemblage is the largest recorded in the Iberian Peninsula. Is more than 2,500 human remains. There are a few stone tools in the assemblage, and we have identified a minimum number of 13 individuals. And also something that is important from the anatomical viewpoint and the research is that all the skeletal regions are represented. All the skeletal regions are represented, but it doesn't mean that the complete skeleton of the 13 individuals has been recovered. No, there are some representation of the all skeletal regions. Something that is important and significant for the analysis, and something that represents actually some quality of the assemblage of the sample that gives some extra information, is that all the fossils come from, as I said, a very restricted area in the site. We can see here a stratigraphic section, and this is the stratigraphic column where the fossils come. It's less than 1 meter deep, and we can see here in this other diagram the directions that we have inferred from the fossils coming. So, because of different criteria, for instance, there are very few final remains, which means that the occupation time was very short. Well, I have to say that the collection fossil, the Neanderthals from Sidrón, are located in a secondary position, which means that the assemblies come from probably from an upper level in the karst, and because a big storm, probably some kind of event that result in a collapse of the karst gallery, all the assemblies fall down into a lower level. And there in the assemblies, in the fossil assemblies, we found, as I said, very few faunal remains. We speak about a short occupation time. There is a low number of lithic industry, but these stone tools can be refitted. What they speak about a single and brief cultural activity, and there are some refitted flake, and bones can be refitted. And also something that is important is that some skeletal parts still are in anatomical connection. We have here an example on the right. So what means that there is a synchrony in the assemblage of the 13 Neanderthal individuals, and probably they all belong to a single Neanderthal group, which gives us the opportunity to explore a number of research lines, also considering aspects related to relationship between a single Neanderthal group. It's just not talking about Neanderthals in general, it's not talking about individuals Neanderthal, but a single group, and the demography and some aspects has been possible to explore. In general, I show you this diagram saying that we have following different line of research, actually trying from the molecular level, because we have contribute and participate quite actively in the development of especially in the first step of paleogenetics and now paleogenomics. We also study at the other different biological level aspect of histology, aspects related with the morphology of the different anatomical systems, for instance, the reconstructing the endocranial anatomical features. As I said, the several aspects related with the Neanderthal population within this single group or the relation with other Neanderthal groups. We will speak about the paleodemography of this human species, and also what is actually my personal major interest is the study and the reconstruction of the evolutionary model and how Neanderthals are related to other human species and how to reconstruct the tree of life of hominins. Let's go to explore a little bit different aspect of these paleobiological studies, and we start with the restoration and the conservation of the fossil. This is a first step. You have in South Africa a large experience in the processes of cleaning and making the fossils available for study. In that particular case, this maxilla originally was covered by a crust that was, as you can see, especially at the occlusal surface, it was just impossible to study. With the help of specialist people, has been cleaned, and nowadays we have this nice maxilla ready for analysis. We started at the beginning also using the virtual techniques for the, let's say, digging some particular fossil. For instance, this block which contains a foot in anatomical connection, or also a spine with several, we can see here several vertebrae and ribs here, the foot, and we try to excavate from this block to separate, to isolate digitally the different elements. But I have to say that unfortunately, this methodology is useful for some aspects of this process. But at the end, we have to use mechanical techniques for removing this crust that is so close to the bone. and the density of these crust and the bone are so close that the digital method are not able to distinguish and to separate properly. So at the end, we have to use this mechanical and classic methodology. Another aspect that we approached with the study of Neanderthals from El Sidrón was to ratify, to say that they are properly Neanderthals. And the first step was just to identify some derived feature that clearly classify these samples as Neanderthals. Here is a simple example of a mandible in which we can see clearly the position of the mental phenomena below the M1, that is a classic derived automorphic feature of Neanderthals. Also something that we will retake later in the lecture, the position, the change in the inclination of the mylohyoid line, which is also a very typical and characteristic feature of Neanderthals. Also in the occipitomastoid complex, there are a few examples with the transverse occipital torus on the clearly suprailiac fossa, the chignon, and several features that distinguish this sample as classic or typical Neanderthals. I have to say that generally speaking, they are classic Neanderthals, but they, as every sample, as every population, as every group, has some particularities, something that is special just for them. But taxonomically, we can speak clearly about Neanderthals from El Sidrón as is well. Another aspect that has been discovered in this sample that is still under study is the evidence, the clear evidence of human treatment of these bones, the cannibalism, that we can see here in this couple of examples, in the mandible, in the basal border of the mandible. We see here these lines which correspond to cut marks, and also in this humerus, this clearly cut mark that has been studied at the electron microscope respond clearly to the typology of the cut marks. So it's another aspect of this sample that has been approach and is still in the particular case is still under study. Something that has some implication and is the basis of another study is the identification of the number of individuals and the reconstruction of these individuals that compose this El Sidrón Neanderthal group. As I said before, we have to identify 13 individuals. There are 7 adults, 3 adolescents, 2 juveniles, and 1 infant. From there are 3 females and 4 males from the adults. And at the time of reconstructing this individual, we have used especially the dentition. Some of the dentition of this individual appear together as the maxilla we saw before, but in other cases the teeth has been found in isolated, so we have to use a series of criteria in order to assemblage the different teeth. And one of the criteria we have used for this is the analysis of the so-called these subvertical groups. As you can see in these pictures, there are in some particular tooth, and especially Neanderthals, appear these kind of groups that when you have together in the proximal facets, the group, they are like fingerprints that can be used to relate one to the other. This has been done. We use another criteria, the size, the shape, even the taphonomy of some of the pieces. But this one, the use of interproximal word facet and the subvertical groups in particular has been one of the aspects which has helped us to join these teeth. One aspect that is important in paleontological collection and especially in paleoanthropological collection is the assemblage of skeletons. especially when they appear mixed one to each other, with the teeth has been possible to make this, to assemble this complete maxilla or mandible. But with the long bones, with the postcranial skeleton, actually is a problem that has not been solved at the moment. And it's a pity because every day more and more the study of the anatomy, the study and the evolution of hominins is approached from the study of skeleton or at least anatomical system, not just isolated bones. But as I said, this is a problem that is not solved in paleoanthropological assemblages. And we are still looking for different methodologies, because genetically I asked to the expert people, and it's not, at the moment, there is not enough information to gather enough markers to identify individual as it is done in forensic analysis. But for the moment, this particularity, this detail is not possible to approach. In the cases that, for instance, because of the stage of maturation, and it is possible to assemblage different bones, as in the case of the juvenile one for Sidrón, in which you can see here, for instance, the diaphysis and the epiphysis and fuse, and all the states of development are similar. Some of these bones appear in almost anatomical connection, that in such a way that it could be reconstruct a partial skeleton of a juvenile individual. In these particular cases, the level of the analysis, the level of the studies increase the potentiality and the quality. And in that case, we approach the study of something that has been approached by other researchers with a great interest, and this is the Neanderthal growth, the pattern of growth of Neanderthals, and the question is whether these human species share with us a common pattern of growth, or we have some kind of specificities in our own species, Homo sapiens. So the comparison of these species has been a background for understanding our own biology. We approach the study of this and compare with a large sample of modern humans and also other Neanderthals. And what we found, well, firstly, we estimate by dental histology the chronological age of this individual with 7.7 years. This is a very important aspect, to have the chronological age for establish a stable reference in front of which compare the development of the different part of the skeleton. One of the problems in paleoanthropology is that we don't know the chronological age. We only know what we call the biological age, that is the stage of development. But in this case, because we studied dental histology, it was possible to establish the chronological age, the date, the age at when this individual died. And with this, we could compare the different systems, and what we found is that actually for most of the systems, the anatomical systems, the growth of this individual was within the range of the modern humans. So generally speaking, the growth of Neanderthal, the skeleton of Neanderthal follow a pattern that is similar to the modern humans. But there are some exceptions that is very, very significant. The first one is that the atlas and the mid-thoracic vertebra, that we can see here, are clearly below the age of the individual, correspond to the stage of 5-6 years, which means that the development of the spine is delayed comparing with modern humans. And also we think that the brain growth was not yet completed in this individual. And we think that this reflects some kind of particular aspect of the physiology of Neanderthals and the ontogenetic energy constraints. which means that because Neanderthal had a really big brain, even larger than ours, and also their body were very robust, so they has to divert a little bit of energy for growing the body and the brain, and so probably the final stage of the brain growth was a little bit delayed comparing with our species. And this is something we found with the study with this skeleton. Other biological aspect has been a classic one is the use of the mouth as a tool. And we found, for instance, some kind of chipping enamel. We can see here, which means that the mouth was used for making some different works, or the interproximal groups, or this kind of scratches here, we can see. And we found that most of them were right-handed individuals, but we also found something that could be is actually interesting. That is, we infer some kind of division of labor by age and sex in Neanderthals. That is, so both males and females has in front of their teeth, as we can see here, some kind of this kind of scratches, which means that they were using the mouth as a third hand. But what we found is that the female has longer cultural striations and also a little bit also wider, which means that they were using their mouth as a third mouth, but probably the function that they were performing was in some way different. And it's because of that we infer that there are some kind of sexual division of labor, and also in the juvenile individuals, the juvenile one in particular has some kind of scratches that we think this fellow was starting to do what the adults made in their life. And also the dental chipping means that probably there was some kind of specialization in the work, in the works that the different individuals in the work were performing. Another aspect interesting in the paleobiology of Neanderthals was the discovery of what we call the self-medication. And this evidence has been found in by two different approaches, both of them studying the dental calculus. And in the first one, just studying the particles that were contained in the dental calculus, it was found that the Neanderthals use chamomile and achillea, that are medicinal plants that nowadays we use. And also it was found that the dental calculus trapped bitumen and small waste, which means that they fit with all the archaeological evidence that the Neanderthals were living in a heavily smoky atmosphere. And also, in a more sophisticated analysis, the study of the the DNA that is enclosed in the dental calculus has been detected from several aspects. One of them is the presence of some pathogens and bacteria in an individual, that is, they have an access in the mouth. It was an infection which produced heavy effects on this fellow. But also we found some sequences that belong to the tree, the poplar, which is a natural painkiller, the producer of salicylic acid, that is the active ingredient of the aspirin. And also it was found natural antibiotic has been produced by the penicillin. All of this evidence comes from the analysis of the DNA that is enclosed in this dental calculus. In the study of microware and the diet in general, there has been also some interesting inferences. For instance, that the Neanderthals, the dental microware, this again, as in the other case, two different ways of analysis. The first one is the study of the dental microware with electron microscopy and some variables. And the conclusion is that the dental wear, and by inference, the diet of this Neanderthal is different in different ecosystems. There are good ecosystem, mixed habitat, and open habitat. The dental wear in this different habitat is different, which means that the way of alimentation, the diet in these groups are different. And this has been confirmed also with a study of the dental calculus, the DNA of the dental calculus, in which has been found, for instance, that the Neanderthals living in the north of Europe, particularly in the cave of Spee in Belgium, they ate the heavily meat-based food with rhinoceros and wild sheep. But in the south of Europe, at the Sidrón cave, which is it was a forest environment, there has not been identified detected meat, but it has been identified mushroom, pine nuts, and moss. I have to say that it doesn't mean that the Sidrón Neanderthal didn't eat meat, but in this particular case, in this particular sample, the major amount of food was composed by mushrooms, pine nut, and moss, and no meat, which means that there are some differences between the different groups. Again, I said confirmed by dental microwear and the analysis of the DNA that is contained in the dental calculus. And this aspect can be related with what we call the Neanderthal phylogeography, which is the differences between the population that were living in the north, which during the glacial period probably went down to the refugia in the peninsula from the south of Europe. And we found in a study the anatomy of the mandible in the Neanderthals from El Sidrón that there was a difference between north and south Neanderthals. These differences has been also found with the study of mitochondrial DNA, and as has been found, for instance, that there was a population turnover in Western Europe during the early stage 3, and this has been recently confirmed with the study of, in that particular case, DNA that is preserved in the sediments of the cave. In this particular case is in the cave Galería de las Estatuas in the Sierra de Atapuerca, also in Spain, where people has found that there was a population of Neanderthals, population 1 here, and then there was a replacement, a change in the population of Neanderthals that it was called population 2, which means that the life and the history and the evolution of the Neanderthal population has been much complex than has been thought before just from the study of the anatomy. The study of the genetic markers are revealing that the history of Neanderthal has been really complex. And as another example, that the study here is before that, the one we saw just now, was the identification in the sediments of the cave, in several caves, both DNA, genetic evidences from Neanderthals, and also from animals. What, in my view, actually, that means that we are living now a radical change in the quaternary science with the capability we have nowadays to recover DNA or other molecules, not only from bones and teeth, but also from sediments. And in my view, we are, as I said, approaching a radical change and what we call a scientific revolution that is being produced at the beginning of this century, in which I like to say that with this scheme that the classic science of prehistory, they study the fossils and the stone tools on the one hand, and on the other, the molecular sciences with the study of ancient DNA, but also from proteomics and other molecules, are interchanging information and producing a new paradigm in human evolution. And in the Sidrón, as I said, we contribute a little bit to this revolution. And some of the aspects we have developed specifically at this site was what we call the clean excavation. So we have developed years ago a protocol for extracting bones in such a way that we try to avoid any contact of our body with the fossils in order to avoid the contamination with our DNA. We can see here a short movie that illustrates what we have developed at the site in order to extract Neanderthal bones fossils without modern human contamination. We are here at the Sidrón. We go into the cave, and we are now in the main gallery. And after 250 meters, then we arrive to a small lateral gallery where our colleagues are working there. Here, for instance, we have a bone. And once we detected the presence of a bone that would be able to send to the lab for extracting DNA, we leave the gallery, and some people wear these specialized clothes with masks, with double control, and we go into one of our colleagues go into the gallery again with this kind of bag. Here again we are in the Galería de los Huesos, and these small bones that was discovered just a little bit is going to be discovered properly. Here we see another double control with sterile globes. And we have here this small piece, this small bone that appears just a little bit. So we complete, the instrument has been bleached, and here is, okay. This small fragment that we can be certainly sure that this did belong to a Neanderthal, because the specific characteristic of this site at the Sidrón, where almost all bones come from Neanderthals, is extracted, put in bags, and then preserved in cold temperature in order to keep stabilize this bone and stop the potential processes that degrade the DNA. We went out of the cave with a sample, and then this sample is taken to in that particular case at the hotel where we have a bridge that is frozen at -20 degrees and sent to the lab, and the process for extracting the molecules has been performed. And one of the major results in this revolution and in our modest contribution to this scientific change and revolution is our contribution to the Neanderthal genome, where samples from the Sidrón were used as a test for controlling and for extracting the long sequences of DNA of these molecules. And as you know perfectly, the general conclusions of this Neanderthal genome was the fact that we recognize nowadays that the process of inbreeding is the hybridization and gene flow is a constant in our evolution and in the evolution of the human species. Let's go to see quickly another movie in which we can summarize the major conclusion from this original study. If we look at the Earth at 150 years ago, we see that the planet were living at least 5, 6, or even more human species. We see Homo sapiens in Africa, Homo neanderthalensis in Western Europe, the Denisovans, Homo erectus, Homo floresiensis nowadays has been defined, Homo luzonensis in Africa perhaps. At 7 or 60,000 years ago, there was a moving outside the African continent, and the Homo sapiens populations occupied what we call the area of inbreeding were living the Neanderthals. And the descendants of these hybrids were dispersing all around the world. And at that time, more or less close to 40,000, the archaic populations, species like Homo neanderthalensis and other, were becoming extinct. And later on, Homo sapiens finally occupied all the corners, all the places in our planet. But the descendant of this hybrid, we carry part of a percentage of this Neanderthal DNA. Most of the human populations not from the south of Sahara populations, we carry more or less 2 or 3% of our genes come from the inbreeding with Neanderthals. And apart from this important aspect in our evolution, in which, as I said, and I repeat, the gene flow is a constant process, not only with Neanderthals and modern humans, but also with Neanderthals and Denisovans and modern humans, and probably modern humans and some archaic species that has not been still discovered properly with fossils. And there are other aspects that are increasing even more our interest in the study of Neanderthal. Let me say quickly what is the basis of this new discovery and the renewed interest. And let's go talk a little bit about the evolutionary origin of Neanderthals. As we comment at the beginning of the lecture, the Neanderthals has a predecessor that can be found in the Middle Pleistocene of Europe, and specifically in the Cima de los Huesos of Atapuerca, where they have a number of anatomical features that speak about the relationship between this Middle Pleistocene and the Upper Pleistocene population. So the Cima de los Huesos are the ancestors of Neanderthals. I think everybody nowadays will agree with this interpretation. But there is something that is really curious, and it's the fact that when it was extracted mitochondrial DNA from the Cima de los Huesos, the result was actually really strange, in the sense that the Cima de los Huesos was clustered with the Denisova, but not with Neanderthals. So there is something that is not matching our ideas, because when has been sequenced the nuclear DNA, it is close to Neanderthals, but the mitochondrial DNA and also the Y chromosome DNA, it is close to the Denisovans. We have, in this way, we have a kind of contradiction that it has to be solved. And I repeat here, when we look at the nuclear DNA and we compare sapiens, Denisovans, and Neanderthals, that's fine. It's what we would expect, and is in agree with the anatomy, with the morphology. Neanderthals and Denisovan may form a cluster, and the sapiens is outside. But when we look at the mitochondrial DNA and the Y chromosome, something happened in the way that Neanderthals and sapiens cluster together, and the Denisovan fall outside of this cluster. So there is some kind of unmatching or contradiction. How can this contrasting signal of mitochondrial DNA and Y chromosome and the nuclear DNA come reconciliate? It has been proposed that in the evolution of these groups, we have modern humans here, we have Neanderthals, we have Denisovan. Some kind of inbreeding put the modern humans into the Neanderthals, and is preserved in Neanderthals only the mitochondrial and the Y chromosome, in such a way that because of that, we have this connection with this monoparental DNA, and the nuclear DNA is recorded the general structure. So it has been proposed, and this is something completely new and very recent, that in sometime in the middle Pleistocene, there has to be some kind of inbreeding between early modern humans, the ancestor of modern humans, and the ancestor of Neanderthals, in such a way that they transfer this sort of DNA into the Neanderthal. What is the meaning of this inbreeding, of this hybridization? What is the consequences for the Neanderthal evolution of this gene flow that happens in the middle Pleistocene? We don't know. We have to study, and once again, from this new perspective, another combining with the classic one, what is the influence of the genome of modern humans into the evolution of Neanderthals? And in this way, and because of this, and because of classic questions, we are now once again re-asking, making the question that what are Neanderthal traits? What are really Neanderthal traits? And what is the direction of the change that we see in the Neanderthal lineage? Classically, this kind of question has been approached by a direct comparison between modern humans and Neanderthals. And we are trying now to enlarge the scope of comparisons and trying to compare Neanderthal, not just in this direct comparison with modern humans, but with other Homo species. And let me now give a quick review of the Neanderthal feature and what could be the direction of change and the pattern of change that we are detecting in the Neanderthal lineage. Just comparing body form and the proportion, we see that the Neanderthal has a very peculiar body form, which is quite different from Homo sapiens. They are short and wide, and this is something that has been probably inherited from the ancestors, but there are some particularities. For instance, the short tibia in the Neanderthal that has some kind of apomorphic expression in Neanderthals. Also, in the anatomy of the skull, we can see, I go here quickly because we are running out of time. For instance, the mid-facial prognathism, that is a very peculiar structure, a very peculiar feature that defines the Neanderthal properly. And we found that this very peculiar feature of Neanderthal is not only specific for the species Homo neanderthalensis, but also has been inherited and developed in the species what we call Homo heidelbergensis, that is the ancestor from the middle Pleistocene in Europe. Also in the mandible, there are some features that are primitive in Homo heidelbergensis and others that are derived, and some specific feature of Neanderthal in the Neanderthals. There are something that is interesting here, that is in the cervical and the lumbar spine. In the Neanderthal lineage appear some kind of hypolordosis. Curiously, the only genes that has been detected in Neanderthal that are specific of Neanderthals and not with the difference with Denisovans and modern humans are related with this kind of hypolordosis or hyperlordosis. So something is happening here in the evolution of the Neanderthals. The pelvis also has probably effect of this lumbar lordosis that has to be explored, and there are also specifically Neanderthals some derived feature of this part of the lineage. In the thorax, the structure seems to be primitive all along the lineage. In the upper limb, there are also some features that has been derived in the beginning of the Neanderthal lineage with Homo heidelbergensis, and also here is in yellow, and also there are some specific features that develop in the final part of the lineage that we call Homo heidelbergensis specifically. And in the lower limb, there are also some features that seems to be developed in the Homo heidelbergensis, and some specific features that are developed just in Homo heidelbergensis. Well, just as a summary, I went very quickly on this because something that is very specific for a specialist. At the end, in our view, what we see is that the Neanderthal lineage has two different phases of evolution. And the origin of the anatomical features are related with systemic changes, not just this an accretion of small changes in the skeleton, small changes in the anatomy that at the end produce a Neanderthal. We think that the changes are correlated, that has been changed in a systemic way. And we differentiate what we call the phase one is the change between an ancestral species and the Homo heidelbergensis species. And in that case, the changes that we have detected are related with the mid-facial pronatism and the hypolordosis. So changes that has to be related with the dorsoventral axis or plane of the organism. And in the second phase, what we find is, first of all, a great increase in the brain volume, reaching the 1,500 cubic centimeters. So there is a process of encephalization. And in this process, we think that there has a head basculation. The head changed the equilibrium with the cervical column, and that moved all this attachment of muscles and the relative position of different aspects of the anatomy. This is, I think, this has to be with the evolution of this evolutionary lineage of the Neanderthals, and within these changes, it has to be detected what could be the effect of the proper evolution of this lineage and what could be the proper evolution, the proper influence of the interbreeding with modern humans. This interface between the local evolution of this population and the influence of modern humans, the ancestors of modern humans, has to be explored in detail because this is completely new in research. To summarize and to finish the lecture, we can see again this in the chronological axis, the different fossils. We think there is the first phase of the evolution of the Neanderthal lineage with a large diversity that has to be explored why there is this variation. It's quite actually high. Probably there is a bottleneck here around the origin of the proper Neanderthals between 300,000, 450,000 with a strong decrease of variation, and then probably there is some kind of diversification within different areas of population, but within the Neanderthal, the Homo neanderthalensis species. So this is a general framework we have developed with the study of this sort of fossil, but we have used the Sidrón as a reference for this. And there are also many questions that has not been solved. Classic ones and the new ones that has been coming from the improvement of methodologies, the improvement of techniques, and the revolution that is becoming from the study of the ancient DNA. And with this, I want to finish and to thank you for your attention, and I hope you enjoyed a little bit, at least some aspect of this lecture. Thank you very much.
Oh, thank you so much. That was fascinating. So I would like to open the floor now to our guests. If anyone would like to ask a question or say something in the chat, you can type it in the chat. I'll read it out, or you're welcome to put up your hand and then just unmute yourself. And you're all welcome to put on your videos now. It would be nice to see some faces also for Prof. Rosas to have a look at who he's been chatting to. So we've got some thank yous for many thanks for the most informative and thought-provoking talk. This is fascinating. Thank you.
Thank you.
Cool.
Thank you very much.
Okay, so if anyone has some questions, you can just put up your hand. I would like to ask something from unknowledgeable perspective about Neanderthals. So you're talking about hypolordosis.
Yes.
Just from the image, I wasn't sure exactly what it meant. You know, the spine looked more upright or smaller at the top. What does it exactly mean?
Well, the hypolordosis means that, as you know,
Can you talk a little bit louder? Sorry, can't hear you. Combination of you hearing us and us hearing you.
okay, can you listen to me now?
Not as well.
Okay, okay.
What about now?
Yeah, now I can hear you nicely. So,
I'll choose this way. As you know, we have in the spine a series of curvature that are needed to maintain our posture, bipedal posture, and one of them is we call the lordosis, the lumbar lordosis, and we have a quite pronounced. Let me go back here. Okay, so we have, like here, in our species, in our anatomy, we have a quite pronounced lumbar curvature in here, and we detect in the Neanderthals, and probably these processes start in the Homo heidelbergensis. This curvature decreased quite a lot, and it's called the hypolordosis. We don't have an explanation for that. We don't have a functional interpretation about why it is produced this change in the structure. It is quite we have proposed, and we are developing now, we are actually aware at the beginning in our group, trying to explore how this is connected with the mid-facial prognatism, that is the projection of the middle part of the face, in the sense that this plane of the body, both in face and in the column, has been modified. Why? No idea. We are trying to understand whether, first of all, whether there is connection between the mid-facial prognatism and the changes in the vertebral column, this first of all, and then whether there are some genes behind these changes that are integrated probably in a very early stage of development in the embryo. And what could be the functional interpretation of this? Again, I repeat, unfortunately, no idea.
sure, that's interesting. So they had better posture than us humans. Okay, then there's a question from Serge. Highly fascinating, thanks. Are there any signs of marine food consumption among the El Sidrón group?
Not under Sidrón. No, there are some evidences of marine food in other Neanderthals. Actually, there is a discussion because it was proposed that the marine food was one kind of reaction for the evolution of our brain, because it's quite nutritious, the marine food, and it has been detected in other Neanderthal groups, but not at the El Sidrón, because actually the El Sidrón cave is close to the coast, but there is a mountain range in between, and probably this group of Neanderthal were living close to the coast, but at the other side of the range, of the mountains. So it's quite possible that they don't have a direct connection. Nowadays, we have to go up and then down to get the sea from the side. So no evidence of marine food in the Neanderthals from El Sidrón.
We have another question from Sally Roper. An early map in your talk indicates that Neanderthals also occurred in the UK. I'm not very up to date with this aspect of Neanderthals there. Any idea how they got there?
Well, the British island has been connected to the continent for many, actually perhaps most of the time in the history of Europe during the Quaternary period, the British island has been connected to the continent, and it is at present when they are islands probably, but the connection between the islands and the continent has been constant and during several phases and periods, and many other animals has been living in the British Island in Great Britain.
Then a question from Kinilwe Molopiane. Love this talk. Are there any signs of trauma in the collection?
There are some. No, actually there are no physical trauma. But what we have found is something that is quite interesting. There is some kind of genetic anomalies that are recorded in the skeleton. Small little changes that probably they don't decrease the viability of the life of the individuals, but they are speaking us about probably the inbreeding within Neanderthal groups, because this sort of pathologies, they are not pathologies, they are variations that are related with genetic anomalies. Not really big ones, the ones that at the atlas, for instance, in the lumbar part of the column, there are also some examples in the clavicle. Little details that speak us about the inbreeding with Neanderthals. And actually we propose that perhaps this small size of the group, high incidence of inbreeding within these groups, perhaps could be one of the reasons of the Neanderthal extinction, in the sense that there is a decrease in the genetic variability that is necessary to keep the evolution working. But no physical trauma, that, by the way, that is quite common in other Neanderthal samples, not in the Sidrón, but this kind of genetic anomalies that are recorded in the skeleton.
Yeah, and you partially answered a question that we got from Penny. Why did the Neanderthals become extinct? So what are other possible things?
Well, the extinction of Neanderthal is one of the classic questions,
yeah.
in the study of Neanderthals. And well, actually nowadays we think there could be a combination of circumstances. One is Europe was changing quite a lot at the time when the Neanderthal became extinct. There was the beginning and the development of a new glaciation. At the maximum was really hard. And probably Neanderthals, against the idea we had previously that they were related with cold weather, perhaps Neanderthals were forest animals, forest people, in the sense that we were adapted to forest, an environment dominated by forest. And at that time, the weather, the climate was deteriorating quite a lot. Probably the ecosystem, the best ecosystem for Neanderthals become destroyed. This is one aspect. The other one is the ones I referred before, is that the genetic variation of Neanderthal, we now know from the ancient DNA is quite low. And we know from the study, for instance, in the Sidrón group, that the inbreeding within these populations and also from genetic data was quite high. And this produced the depression by inbreeding. And there is not enough variation within this population for a healthy evolution, let's say in that way. And also perhaps the arrival of modern humans into Europe at that time was just the final, I don't know what is the English for that, it's the final aspect that killed the population. So at the end is because there is a strong ecological competitor, not because they physically killed a Neanderthals, there is no evidence of violence, there is no evidence, but because there was perhaps a very strong ecological competition. So climate, genetic variation, and ecological competition, the combination of this may explain the extinction of Neanderthals.
And then just a little bit of clarification. I'm sorry I was a little bit late. What is the context in which these skeletons were found?
The context.
Yeah.
Well, as I was explaining a little bit, all the Neanderthal remains from El Sidrón appears in a very small cave gallery in a very restricted area, no more than 6 square meters, and we interpreted that all of this assemblage, the remains that we recovered from these 13 individuals, actually come from an upper level of the karst system. As you know, caves are organized in several floors, levels, and probably the original assemblage was buried in an upper level, and because a very strong storm or something happened that produced the collapse of this gallery, and all the assemblage become deposited in a lower level, the site that we have excavated. So they are in a secondary position.
Then we have a question from Chichen. Have we found any records that show their habitual behaviors? For example, were they migrational like that's found in South Africa, or did they stay in one place for long periods?
No, we could not find evidence for this kind of movement, because we were trying to study the strontium isotope. That is a good marker for this, but we found something that is really interesting. And with the study of mitochondrial DNA, what we found is that all the males individuals share the same mitochondrial DNA. By contrast, all the females has a different kind of mitochondrial DNA. And what we have interpreted with this is that probably Neanderthal practiced what we call patrilocality, which means that the males stay in the parents' territory area, and it was the females, perhaps at the adolescent time, that changed the group. So the group changed young females into another group, and this while the males stayed in the parents' territory. This, combining with other evidence, for instance, the procedence of the raw material for the stone tools, and other evidence, speak about the Neanderthals stay most of the time in a very restricted geographical area, and they don't move to other territories. Actually, there has been a comparison between Neanderthals and modern humans, because it has been proposed that modern humans, one of the differences of modern humans was that they travel quite a lot, and the raw material traveled quite a lot, while the Neanderthals stayed always in the same place, most of the time.
That's interesting. Cool. Yeah, we've got lots of thank yous and really fascinating. And that's great. Are there any other last questions before we close up?
Tammy, I'd just like to say a really big thank you to Prof. Rosas. I think that was absolutely fantastic. It certainly given me a whole new insight to what's going on in research in this particular field at the moment. And I think it's quite exciting, and I certainly we're going to be following it with keen interest going forward. So thank you very much.
Thanks to you.
Thank you so much. Cool. So we have recorded this talk, and so if anyone does want a recording, they can email me. I've just put my email in the chat, and we will hopefully, when we get our YouTube channel up, that we'll put it up on YouTube as well. But yeah, I'd like to again thank the Spanish Embassy in Pretoria for helping this come about, and thank you so much to Prof. Antonio Rosas for giving this absolutely fascinating talk. Thank you.
Thank you very much. Thanks.
Thank you. Cool. Okay, I'm going to stop the recording, but you can stay and chat a little if you want.